Hi guys, welcome to my YouTube channel Unique Bio Classes based on NCERT syllabus for PU first and second year. If you like my channel, please subscribe, like and share and hit the bell icon so that you can get notifications as soon as videos are getting uploaded. Okay guys, we should start today's video. In our previous video we have discussed about the test cross which comes under monohybrid cross. Okay, even test cross can be used for dihybrid cross also, which we are going to discuss today. In our today's topic, we have inheritance of two genes. It is also called as dihybrid cross. This inheritance of two genes you will find on page number 78. Okay, on page number 78 in NCERT textbook you will find this. So about this dihybrid cross or inheritance of two genes, I have prepared a note of it. If you find some information regarding that, inheritance of two genes or dihybrid cross, it is a cross between two homozygous individuals differing in two pairs of contrasting characters. Okay, this is a cross between two homozygous. It is not heterozygous, two homozygous individuals which differ in two pairs of contrasting characters. Whereas in inheritance of one genes, what we said, cross between homozygous individuals differing in a pair of contrasting characters, differing in one pair. Here it is two pairs. Okay, now two pairs of contrasting characters are there. Now example for that which has been chosen by the Mendel are two pairs of contrasting traits where one was seed color and another one was seed shape. Okay, if you remember about 7 contrasting characters in that we have given, seed color was round, yellow and green, seed shape was round and wrinkled. So there are two contrasting characters which he has chosen. It was round seed with yellow color, wrinkled seeds with green color. This was the example which he has used for explaining dihybrid cross. About this you have on page number 79, okay, in your textbook. This is that cross. Okay, here it has been mentioned as P generation. It is parent generation. It is having round yellow, round color, round shape, yellow color. For round it has been given it as capital R, and for yellow it has been given it as capital Y. Similarly, wrinkled seed and green color, small r and small y. Okay, so with this is the genotype, whereas round yellow or wrinkled green is phenotype. About this cross, I shall cross that one in my note, so we shall see how it will appear. Okay, fine. Now we have chosen one parent as round yellow and it is crossed with wrinkled green. Okay, so this is something like this, and this one is P generation or parent generation, and it is also called as phenotype, right? This is the phenotype of parent generation. What is that phenotype? Physical appearance, outer appearance, morphological appearance. Round seed, yellow color, wrinkled seed, green color. Now in this, one can be female, another can be male, whichever you want you can give. Okay, for round we have to give genotype. Now genotype of round, what it has been chosen? It has been chosen as capital R, and for yellow it has been given it as capital Y. Okay, for wrinkled green, what will be the genotype of this? Small r. For green it has been given it as small y. Now already we have discussed, if we are giving mentioning with the capital R, then it is dominant. If it is we are mentioning with a small letter, then it is recessive. Now in this it is very clear, round yellow is dominant, wrinkled green is recessive in condition. Okay, fine. Now next what we have to give? Gametes. Gametes of what? Gametes which comes after this parent generation. So gametes will be produced after meiosis. So gametes will be always haploid. So we'll have capital R and capital Y. One round and one yellow gene you will find in this gamete. One more copy of it, if you want, you can write. Okay. Next one is small r, small y. Okay, one from here and one more, same as if you wanted to write one more copy of it, you can write. So after meiosis gametes are formed, and then after gametes there is formation of gametes, there will be a fertilization. So fertilization results in formation of F1 generation. Okay, this one is F1 generation. Now F1 generation, what you will get here? Capital R, capital Y, small y. Okay, capital R, capital Y, small y. We are getting like this. Now in this we have said homozygous individuals. You can find these are homozygous, not heterozygous. Now what we got in F1 generation, that is heterozygous. Now what will be its phenotype? Genotype we get capital R, capital Y, small y. What will be its phenotype? As you know, capital is dominant, so capital will express. So capital R we have here. So what is its phenotype? It is round, and capital Y is yellow. Okay, so these two, it has been round and yellow. First generation we got this, right? Okay, now after this what we have to do? We have to write the gametes of F1 generation. Fine, we shall mention here gametes of F1 generation. Okay, gametes of F1 generation. How many gametes you will get? First one. While writing the gamete, students will get confused always. We'll get 4 different types of gamete. How we'll get 4 different types of gamete? You have to take this one as one set and Y as another set. You don't mix it up for all. First we'll take capital R. It is crossed with capital Y. Capital R with capital Y. Then capital R with small y. These are the 2 gametes which we get. Capital R, capital Y, then capital R, small y. We got like this. Then what is left? Small r. Small r, capital Y. Small r, capital Y, and small r, small y. We'll get like this. Okay, these are the 4 gametes which we'll get from one parent. Now, here what we'll say? Here it will be self-crossed or self-pollinated. When it is self-pollinated, what you will get? You will find 2 types or 2 sets of gametes. This is one set of gamete, another one more set of gametes. Total 8 gametes will get here. But because we have written only one parent, so we have mentioned only 4 gametes. So now what we have to do? We have to draw the checkerboard so that we can get the all 8 gametes. Here, because we are having 4 gametes, we have to draw 4 rows and 4 columns. Okay, this is 1, 2, 3 and 4. Okay, this is the checkerboard which we are going to draw for dihybrid cross. Okay, in this dihybrid cross you will get clearly one side we are giving it as female, on the other side we are giving it as male. Now from one parent we got 4 gametes, which are those: capital R, capital Y, 1, capital R, small y, then small r, capital Y, then small r, small y. Okay, these are the gametes of one parent. So one more set of parent, after self pollination one more set of parent will be there. So its gamete you will have same 4 gametes. Okay, capital R, capital Y, then capital R, small y, then comes small r, capital Y, then small r, small y. Okay, this is how you are going to write the gametes in row and a column. Fine. Okay, now we have to cross this. How you will do? Here you will take capital R from here, then capital R from here, capital Y, capital Y. Okay, then you are taking this gamete and fusing with this gamete. Okay, then comes capital R, capital R, capital Y, small y. You will take this gamete and fuse with this capital R, small r, capital Y, capital Y. Okay, next capital R, small r, capital Y, small y. This is fused with this. Similarly, you are going to fill the other rows and columns. So this gamete when it is fused with this capital R, capital R, capital Y, small y. Then capital R, small y, small y. Then comes capital R, capital Y, small y. Capital R, small y, small y. Same, you are taking this gamete and fusing with each one of it, you will get this. Similarly, we shall fill the other gametes also, fuse the other gametes. Like you can take capital R, capital Y, capital Y. Capital R, capital Y, small y. Small r, capital Y, capital Y. small r, capital Y, small y. Okay, you will get these generations. Next, capital R, capital Y, small y, capital R, small y, small y, small r, capital Y, small y, small r, small y, small y. Okay, so this is how you are going to fill. Hope you understood how to fill this. First, you are taking one gamete fusing with the other gamete. You are writing the genotype, something like this, and filling it. Okay. Now, if this is the what it is, F2 generation. This one is F2 generation. This complete whole set is F2 generation. How many F2 generation we have got here? We have got 16. Okay, 4 plus 4 plus 4 plus 4. Total 16 genotypes we have got. First, we shall write what are its phenotype. As you know, if somewhere capital is there, capital, then it is dominant, then it will express. So this one is round yellow. I shall write it here. It is round yellow. Okay, next, round yellow. This is also round yellow. If one capital is there, capital R or capital Y, then it is round yellow. Then this also round yellow. This one also round yellow. Fine. Now this one, we have capital R and capital Y. So this is also round yellow. Here capital R round, both are small y, so it is green, round green. Next, we have capital R round and it has capital Y also, it is yellow. Now here you have capital R round, it has small y, it is green. Capital R round, capital Y yellow. Capital R round, capital Y yellow. Small r wrinkled, capital Y yellow. So we got wrinkled yellow here. Next, small r wrinkled, capital Y yellow. Next, capital R is there, so it is round, and capital Y is also there, yellow. Capital R round, small y green. Small r wrinkled, capital Y yellow. Then here you have small r wrinkled with the small y green. It is wrinkled green. Okay, you have to fill something like this. If you don't write this phenotype, then also it's okay, but you must know what does the genotype will represent. Okay, now we have to write phenotypic ratio. Okay, I shall show you here. Phenotypic ratio. Phenotypic ratio of dihybrid cross, very important it is. Okay, what are the phenotypes which we have got? We have got 4 different types of phenotype. One is round yellow. Okay, all are round yellow. Next we got round green. Okay, we got one more round green. This is the second phenotype. Then we have got wrinkled yellow and wrinkled green. 4 different phenotypes we have got, and how many are this we have to see. Round yellow, 1, 2, 3, 4. Okay, then comes 5, 6, 7, 8, 9, 10. Okay, so if you count, sorry, it should be 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. So total 9 round yellow we have got. 9 of those what? Round yellow. Okay, then this we have counted. What mark I have given, that has been counted. Leaving that, we have got here round green 1, one more round green, then here you will have one more round green. So I shall make like this. Okay, 1, 2, 3. 3 round green. Okay, now we have counted this. Next comes wrinkled yellow. wrinkled yellow 1, 2, 3. So 3 of those wrinkled yellow, and lastly only one has been left, that is wrinkled green. 1 wrinkled green. Okay, so what is the phenotypic ratio? Phenotypic ratio is 9:3:3:1. This is the phenotypic ratio of dihybrid cross. Whereas of monohybrid cross, you will get a phenotypic ratio as only 3:1. Here it is 9:3:3:1. So you will find a parental type and non-parental type also. Round yellow and wrinkle green are the parental type. Round green and wrinkle yellow are the non-parental type. They have been mixed up and we have got these varieties. This is the phenotypic ratio. Okay, so if you find here in your textbook, it has been shown something like this. See, gametes of one parent, gametes of another parent, and it has been crossed, and it has been shown something like this. And here they have given phenotypic ratio 9:3:3:1. If you wanted to count genotypic ratio, how you will do that? Genotypic ratio, it's quite confusing, but then too we shall try to solve here. Okay, genotypic ratio. As we have got genotypic ratio in monohybrid cross, that was 1:2:1. Here we shall see what are those and how are those. Okay, we have to first see, we have got first one round yellow, capital R, capital R, capital Y, capital Y. How many of this genotype we have? See, we have only one. Another we have capital Y, small y. Like that you have, which is not exactly similar. Same genotype, do you find somewhere? No, we won't find. So this is counted as 1. Okay, because it has been counted, I'll mark it as something like this. I am crossing it. You don't cross it, just I am showing that just it is to know that we have counted this one. Next, capital R, capital R, capital Y, small y. Similar to this, how many genotypes you have? This is 1. This is 2. Okay, then that's it. Okay, this we have counted. Second one is 2. We are going to write like this. Next, we have this one, capital R, small r, capital Y, capital Y. Okay, similar to this, how many we have? This is 1. Okay, then capital R, small r. Okay, we have this one, 1, 2, 5. We have only that 2. Okay, we have got 1:2, 2:2, 2:2. Then capital R, small r, capital Y, small y. So we shall see this. Okay, capital R, small r, capital Y, small y. This is 1. One more you can find. This is 2. And then comes, this is 3. Then this comes 4. Okay, next one is 4. Like this. Which we have crossed, we have counted. Now this has not been crossed. Okay, capital R, small y, small y. How many you have? This one, this capital R, small y, small 1, this one 2. Okay, okay, that is 2. Next comes capital R, small y, small y. Sorry, it should be, this is only 1. Sorry, by mistake I wrote it as 2. This is 1 and 2. Then comes this 2. 1. Then these left, small r, capital Y, capital Y. How many you have? No, only we have 1. Then comes small r, capital Y, small y. We have this one, 2. And this one is the last one, small r, small y, small y. We have 1. So this is the genotypic ratio. 1:2:2:4:1:1:2:1:2:1. This is the genotypic ratio. You don't need to scratch all this and do all this. Just if you have marked what I showed, these triangles, what I have shown earlier for phenotypic ratio, if you show that one, it is enough. But genotypic ratio, if you are going for entrance exam, then you can give this genotypic ratio. If not, in theory it has not been asked, nor it has been given in your textbook, so you just be free from that. If you are very thorough in writing the genotypic ratio, then you can write. If not, don't try to attempt to write. Phenotypic ratio is compulsory, you have to write, and you have to write which are 9, which are 3, which are 3, and which is 1. Okay, so this is how we are going to write about dihybrid cross. This is the schematic representation, very important schematic representation. 5-mark question it is. Explain inheritance of 2 genes or explain dihybrid cross, something like that they will give. Here I have given only definition with example and schematic representation of that with phenotypic and genotypic ratio. But during the time of exam you have to explain if they have asked for explanation. Okay, explanation is given in your textbook, you can cover that part. And in inheritance of 2 genes we have one more law, third law. That third law is law of independent assortment. If you remember this one, okay, we have discussed about principles of inheritance or laws of inheritance. First one was law of dominance, second one was law of segregation, third one is law of independent assortment. So in dihybrid cross you can get this law of independent assortment also. They can define law of independent assortment and explain with an example. So for definition you have it on page number 80. Okay, in your textbook here you will find the definition. When 2 pairs of traits are combined in a hybrid, segregation of one pair of character is independent of the other pair of character. Okay, we are taking 2 pairs of traits. Traits means what here? Characters. When 2 pairs of characters are combined in a hybrid, say combined, here 2 pairs of traits are there. Okay, it is combined in an hybrid. This one is the hybrid. Okay, segregation of one pair of character is independent of other pair of character. Now one pair of character is what? Round yellow. Another pair of character is what? Wrinkled green. Now that segregation, that separation is independent of each other. Both are not dependent on each other. How it is so? I should explain it here. Fine. Now what we said, capital R, small r, capital Y, small y. This we have got hybrid in F1 generation in the previous cross, which we have drawn just now. Now, what gametes we have said? Capital R is combining with capital Y, capital R with small y, small r with capital Y, small r with small y. We said something like this. Okay, now this capital R is representing round, small r is representing wrinkled. Now this character is independent of each other. Now both are not dependent on each other. Like capital R is free of small r is free of capital R. If both were not independent, then always you would have found both were together. But you can find here capital R is with capital Y, not with small r. Okay, if we have got capital R, small r, then capital Y, if these both were together, then both are dependent. But here we didn't find something like this. Small r is moving separately, capital R is moving separately during the formation of gamete. Okay, why they are moving separately? Because they are independent of each other. Okay, now capital R is not always mixing with the capital small r, nor capital R is always mixing with the capital Y. Capital R is with capital Y, capital R is with small y. 50-50% chances are there. Is it? Capital R, it can fuse with either capital Y or small y. Is it? So capital R, either it can with this or either it can fuse with this. Chances are there, 50-50% chances. This chromosome or this gamete can fuse, this sperm can fuse with this one. So you can get capital R, capital Y or small capital R, small y. 50% chances are there. This one can, this capital R can fuse with this, or capital R can fuse with this y. And this small r, it also can fuse with small y. Is it? Chances are there. This small r can fuse with capital Y. These chances are also there, which means all are independent of each other. If they were not independent, we would have not got these 4 different varieties of phenotypes what we have mentioned earlier. Okay, we would have not got these 4 different types of phenotype. Just for example, round is dependent on yellow. Where round is there, yellow will go on that side. Did we found that? No. If it was so, we would have found only round yellow, not round green. Parental type is round yellow, we would have found only that. But here we have found, other than the parental type, we found round green, wrinkled yellow. is it? So that is why, because they are independent of each other. If not, if both were dependent, means round yellow, wrinkle green, we would have found only 2 phenotypes, but it is not so. So that's why we have found 4 different phenotypes. Okay, so for this explanation, okay, and regarding this explanation, you have it on page number 5080, and law of independent assortment with the definition and with the explanation you have here. The schematic representation will remain the same as we have shown earlier, something like this. Same schematic representation with phenotypic ratio, just explanation get differs for law of independent assortment, which you have here. If they ask for dihybrid cross, then definition is here with the schematic representation with phenotypic ratio, and you have to explain that one regarding inheritance of 2 genes. Okay, very important question, 5-mark question, law of independent assortment, inheritance of 2 genes, or dihybrid cross. Okay, if you have any doubts, any queries, please comment it in the comment box, and we shall meet in our next video. In our next video, we shall discuss about incomplete dominance. Okay, stay safe, stay healthy.